Vanicam Liam, this is Liam in for bear. A siRNA drug silenced its target 90% in a dish. In a mouse, it did almost nothing. The team looked for a better target for months, but they were diagnosing the wrong failure. The drug was reaching the cell, then the cell was eating it alive. Can you explain it, Bear?
The siRNA worked. In the dish, it silenced its target 90%. In the mouse, it did almost nothing. The tumors kept growing. The team spent months looking for a better target, but they were diagnosing the wrong failure. When they finally tracked where the siRNA actually went, it never reached the cytosol. The cell swallowed it into a membrane bubble, acidified that bubble, and digested the drug before it could act. A lipid nanoparticle delivers siRNA into cells, but the cell immediately packages it in an acidic bubble that digests it. The siRNA that works in a dish does nothing in a mouse. How does the ionizable lipid solve this? After injection, the nanoparticle is taken up by a cell through endocytosis. The cell wraps its membrane around the particle and pinches off a sealed bubble called an endosome. That bubble is the default fate. Most cargo never leaves it. Proton pumps in the endosomal membrane push hydrogen ions inside, dropping the pH from 7.4, blood pH, down to around 5.5. That acidification is the cell's way of preparing to digest the contents, and for most cargo, digestion is exactly what happens. The ionizable lipids head group carries an amine, a nitrogen that can pick up or release a proton. At blood pH 7.4, the amine is neutral. The particle circulates without sticking to proteins or cell surfaces, but drop the pH to 5.5, and that amine grabs a proton and gains a positive charge, now cationic. The positively charged ionizable lipids interact electrostatically with the anionic lipids in the endosomal membrane. The opposite charges attract, destabilize the bilayer, and the membrane tears open. The RNA cargo dumps into the cytosol, where the RNA interference machinery lives. Only a single-digit percentage of the internalized cargo actually escapes, 1 to 2%. That sounds like failure, but it's enough because even that thin stream reaches the RNA interference machinery intact. And it's what separates a working LNP from an inert liposome with the same cargo. Here's the comparison. 2 nanoparticle formulations, identical siRNA. LNPA uses a conventional neutral lipid. 48 hours later, 8% silencing, most RNA digested in lysosomes. LNPB uses an ionizable lipid, 84% silencing. Same cell, same cargo. 1 charge switch. Without the pH-responsive charge switch, the particle enters the endosome and stays there. Its cargo is digested. The lipid nanoparticle is, essentially, a vehicle with a pH-sensitive lock that only opens inside the trash bag. The endosome is the rate-limiting step. The ionizable lipids charge flip, neutral in the blood, cationic inside the endosome, is what opens that step. That is what turns an inert liposome into a working RNA drug.
Here's what the mechanism demonstrated. The ionizable lipid carries an amine, a nitrogen that reads pH in the blood at 7.4. It stays neutral. Neutral means it doesn't bind proteins or cell surfaces. The particle circulates safely. Inside the endosome, proton pumps drive the pH down to 5.5. That amine grabs a proton, flips positive, and the now cationic lipid attacks the endosomal membrane electrostatically. The membrane destabilizes. The RNA cargo dumps into the cytosol. Only 1 to 2% escapes, but that is enough because the RNA interference machinery is extraordinarily sensitive. The key trade-off, pH-responsive design, solves the delivery problem, but leaves the escape fraction as the remaining bottleneck. Every generation of ionizable lipid optimization is really asking, can we push that 1% higher without losing the neutral at blood pH behavior that makes the particle safe? Your turn. Take this prompt into Claude and push on the escape fraction problem.